So we all know what aging looks like. It's essentially our body deteriorates over time, leading to a variety of diseases. And we've been looking at aging for thousands of years, trying to understand, is there something we can do about this? Because the end of life, for many of us, is not a pleasant thing, and it's certainly not something you would want your children to have to worry about if you didn't have to. And so it's been my quest for the last 25 years, at least professionally, to try and understand the answers to some really big questions. Why do we age? Is there something we can do to slow it down? If you slow it down, will you live a longer and, importantly, healthier life? It's all about health. And finally, a question that we helped answer just yesterday, which we published in Nature magazine, which I'll talk about today, is aging even reversible? Now, it's a very important time for the entire planet to understand aging. And the reason is that in 2018, for the first time ever in our species history, the number of older people over 65 outnumbered the number of people who are less than 5 years old. And that trend will continue to go in that direction. And we're already seeing in many countries across the world that the burden, both socially and economically, of looking after older people who are no longer productive is an economic burden that is really crippling. And the older the population gets as a percentage, the harder it is for young people to be able to cope, to look after their grandparents and their parents. And so what we're trying to do in my lab, and I also speak on behalf of 100 or so labs around the world who do similar research, we're trying to understand, can we compress the last few years of life that are sick into a very short period? We've been working actually in animal studies in the lab for at least the last 20 years, showing that we can quite easily extend the lifespan of mice and rats and even dogs, and make them live healthier right up to the very end until they die quickly. So that's the goal. It's really not to keep us in nursing homes and being sick for longer. We're not extending old age. We're doing the opposite. Our goal is to extend youthfulness so that we can perhaps live to 90 or 100 and towards the very end still be productive members of society and playing whatever sport you want with your grandkids and great-grandkids. And this talk of living forever is really not something that I'm worried about or even working on. It's more about how do we address the problems that we face right now. And the money that we could save is in the trillions of dollars each year across the planet. In the US, it's estimated that just reducing one major disease by 10% would save in the coming decade at least $3 trillion. So this is real money that we could save that could be put to addressing other problems that we have, improving education, combating climate change, and other things. And in terms of population growth, most countries are already stabilizing in their population. And ultimately, the goal would be for population to stabilize and be replaced at normal levels. And you might be thinking, well, David, if you slow down aging, we're going to be overwhelmed with people. That's actually not the case. The number of older people is not that many if you do the math. So really, let's talk about medicine and what the future may look like in the near future. And also, as Shabana said, I'll talk about what we can each do in our daily lives to give us the best possible healthy lifespan. So I work on aging, and the problem is that there are a lot of companies on the internet, a lot of people who really don't have a degree in this field who claim that they've quote-unquote solved aging. Drink this, eat this antioxidant. And the problem is that it's very hard to know what is real science and what isn't. And there's such a big difference between 99% of what is out there, which is mostly wrong or exaggerated greatly, versus what we do in my lab and my colleagues do, which is, as I mentioned, on the cover of leading scientific magazines. And there have even been 2 Nobel Prizes awarded for research that is relevant or based on aging studies. So let's talk about what actually is real and what might actually work, and cut through all of the other noise. So this is an interesting graph that I wanted to share with you. Often we think that we've reached our maximum lifespan as a society. This graph shows that that's not true. What is plotted here is the country with the maximum, the longest lifespan expectancy. Okay, so it started off, there was Australia in there, where I come from, for a while. But right now it's Japan who has the longest average lifespan. And what you can see with this graph is that over the 20th century and continuing to today, there's a very linear and predictable increase in human longevity. And every time these groups, which you can see on the right, have said that, okay, we've reached the maximum, we blow through that glass ceiling, and we keep adding years to life. But they're not all healthy years. We're very good at preventing heart disease. We can lower blood pressure. Cholesterol drugs have been largely successful. We have heart surgery. So we tend not to die as much as we used to from cardiac reasons, but the brain still ages at a normal rate, and we don't do much about that. And so what we're ending up with is an increase in dementia, and one of those is Alzheimer's disease. So our approach is to treat the entire body with medicines and lifestyles that will keep every part of the body healthier and more youthful. So today, most developed countries have a lifespan around mid-70s to 80, which is great. It's certainly better than it was 100 years ago, and none of us would go back to those days before antibiotics, and average lifespan was 65, 70, and we would retire and die a few years later. We're on a path to having a couple of scenarios. One is that we'll live longer, but we'll still spend a large percentage of our life in decline and sick with many diseases and being frail. The other alternative, which is on the bottom here, if we're successful with this research, is that we will live longer and healthier, and that sick part of our lives will be much shorter. So whenever I talk to politicians or business leaders, or even people I meet, I won't say on the street because we're in a pandemic, but most people think of aging as something very different than cancer and heart disease and Alzheimer's. But actually, that's not the case. Aging is nothing special. In fact, aging is by far the largest cause of all the diseases that, well, not all, but most of the major diseases on the planet. And because we've gotten rid of all the easy stuff, we've largely improved child survival. In middle age, we tend to do quite well, but it's the end of life where we haven't been successful. And as I mentioned, the main reason is that we've only been addressing one disease at a time. And to live substantially healthier and longer, you need to address aging itself, which drives these diseases. Just last year, the World Health Organization approved or announced that they would declare aging as a disease, a treatable medical condition. So why is that important? Well, one is that perhaps federal agencies, governments will change the law so that aging can be treated by your doctor. Right now, if there is a medicine, and we think that there might be some medicines already approved for other diseases that could lengthen lifespan, right now your doctor can, but typically does not prescribe these medicines for your long-term health unless you are already sick. An example of that drug is metformin, the frontline, often the first medicine to be given to a type 2 diabetic with high blood sugar. But there's a lot of evidence from looking at tens of thousands of people who have taken metformin that they also are protected against frailty, cancer, and Alzheimer's disease, among other things. But your doctor cannot typically prescribe metformin. Now, there are some countries where metformin is available over the counter, which I think is a good thing because it's a very safe drug. But here in the United States, for example, it's largely inaccessible unless you already have type 2 diabetes. The other reason for declaring aging a disease that's important is that it would lead to more investment in research and drug development to treat and prevent these diseases. I mean, if you are biologically 30, even though you have 50 birthday candles, you're much less likely to get cancer or die from a heart attack. And you might say, well, how would I know if I've got my 50th birthday how old I am? I'll tell you in a minute that we can now very accurately measure within just less than 5% error how old you actually are, not by how many times the Earth has gone around the sun. So what I'm going to do in the next 20 minutes or so is tell you really what is going on during aging, we think. Can we slow it down? And can we reverse it? Now, about 10 years ago, 7 if you go by this paper I'm showing you the figure of, we as a field of aging researchers declared that we'd uncovered what we thought were the most important causes of aging itself. And there are about 8 or 9 of these things that happen to us as we get older. And hopefully you can read them, but there are things such as DNA damage, the loss of the ends of chromosomes here, called telomeres. There's deregulated nutrient sensing, so you get diabetes. Mitochondrial dysfunction. Mitochondria are often referred to as the battery packs of our cells. They give us energy. Without mitochondria, we'd all be dead in about 30 seconds or less. It's like taking cyanide. Cellular senescence here, showing with this leaf. Really what that means is that there are cells in our body that act like zombies, and the older we get, the more they accumulate in our body. And they seem to drive cancer as well as diabetes and other problems during aging. And there are a lot of scientists who are working on this in the hopes that one day we could kill those cells and so that the body could be healthier. Other things include stem cell exhaustion, and we lose the ability for parts of our body to communicate to each other. The brain is telling the body to be young, and some organs will, such as the liver, will also tell the rest of the body to be young, and we lose that communication as well. But what I want to talk about today is a new area that's, really emerging as a major cause of aging. And the reason it's exciting is that it seems like it could be reversible. And that's this one here in the darker green. It's written here as epigenetic alterations. Essentially, epigenetics is the description of how our body turns genes on and off. And for each cell, we need a different pattern of which of the 25,000 genes are switched on versus off. And what we find is in young people, cells are beautifully controlled. If the genes are a piano, they're being played perfectly in a concert. But as we get older, what we see is that the pianist is making mistakes and playing the wrong keys, and it's starting to sound like a terrible cacophony of sound. And if you're listening to this, you probably want to put your fingers in your ears. What we're looking at is trying to understand what drives that process, what makes the cell read the wrong genes as we get older. And is it possible to do a reset? Can you get the pianist to play the beautiful music of youth? And as you'll see, I think the answer might be yes. So this all started with what I call the information theory of aging. Essentially, when we're born, or even when we're fertilized as an egg and a sperm, there's information there. Now, there are two types of information in our bodies, primarily. One is the genetic information encoded by our DNA. We call this the genome, and we hear about it quite often in the news. And it's led to a revolution in biology. Of course, our ability to read the genome now is quite dramatic. A student in my lab could read your genome in just a couple of days on a device that looks about that size. So we've gone from $1 billion to a couple of $100 to read a human genome. That's one type of information. The other type are the regulators of the DNA, the genetic regulators, what I was just describing, that make up the epigenome. And these are mostly proteins and chemical modifications of those proteins, and also chemical changes to the DNA. Now, these are very important because every cell essentially has the same DNA. So how do we have brain cells? How do we have skin cells? It's the epigenome, the pattern of which genes are on and off that determine which cells will become skin and which cells will become nerves. And that pattern has to be maintained in some people for over 100 years. The problem is it's not maintained perfectly. So a good analogy is a CD or a DVD for those under 20. I should explain, we used to use these to store music. They're a good analogy though, because the information that's on these discs lasts for many, many years, probably 100 years if you look after it. But these things used to get scratched pretty easily. And when they're scratched, the player would not work very well. And this is similar to what happens to our DNA. So the DNA would be the music, and the scratches would be the epigenome, the inability to read it. And the question that we've had is, how do these scratches occur? And can you polish those discs to get back the beautiful music? This is more similar to what's really going on. This is the nucleus of a cell on the left, a young cell. The genes are these red dots being read here. This is RNA coming out of the gene. And this line, these loops, are the chromosomes. And you can see some genes are on, the red dot, and some genes are kept off by bundling up the DNA in these protein bundles. We call this chromatin. What happens over time that we know for sure is that this beautiful pattern in a young cell that gives the cell identity and function will be lost over time, so that now the old cell is turning on genes that were once off, and vice versa. And these old cells do not function very well, and we think give rise to diseases. Wouldn't it be something if there was a memory of this pattern that could be reset, so that you could take a complex tissue such as the eye and reprogram it, press a reset switch to turn it into a young tissue again, so that it would heal and function as though it was young again and we were young again. Now, one of the things that we've discovered is DNA damage, and the process of repairing that damage drives this epigenetic change, this epigenetic noise, we can call it. And anyone who's spent a lot of time in the sun will know that DNA damage will cause aging. And we think this is the main reason. Not so much the genetic changes, not so much the mutations, but actually the epigenetic changes, which is good news, because if we have lots of mutations in our cells and we lose the DNA sequence, the genetic information, it's extremely hard to restore that, to find it again. In fact, once you've lost all the copies of the genes, there are 2 copies in most cells, once you've lost that, you cannot get it back without very complicated technology. But if it's just the regulation of those genes, then you could imagine that you can reset it. So this is good news. So DNA, of course, isn't just flopping around our cells. It's 6 feet long in every cell. It needs to be packaged very tightly, but also very accurately. And this is nylon, but it's referring to DNA as this cord. And we have these proteins called histones that the DNA is wrapped up. And where it's wrapped up very tightly in these bundles, the genes are switched off. And when they loop out, those genes become accessible to proteins to read the gene. And that's how our cells control which genes are tightly bundled and off and which ones come on. And of course, these are happening all the time. Every time you have a memory or you eat a meal, the cells will respond and change which genes are on and off, but not so much as you might think. Because if you change the structure too much, the cell could become a cancer cell or it could become old. Mostly what happens is that these bundles stay the same for your whole life until you become old. And there are some processes that control that. One of these processes is called methylation. Methyl is just a type of chemical. It's a carbon atom with 3 hydrogens. And the cell attaches these methyl chemicals to the DNA and to these histone packing proteins. And that helps establish which genes will stay off and which will come on. Interestingly, these what are called methylation patterns, while they're beautifully arranged when we're young, they do change in predictable ways as we get older. And as I'll show you, we can use these patterns as a clock. So here's an example of that. Well, on the Y left axis is biological age in years, and on the bottom is the chronological age, so your birthday candles. And what the field has discovered, I need to give credit to my colleague Stephen Horvath. Sometimes we call this the Horvath clock. What we've seen is that these changes in these methyl groups on the DNA can be read by the student very quickly. It can take it's a couple of days to get the answer. Some people, typically those people who have bad genes or have not looked after their bodies, will be older than their actual chronological age. Some people will be average, and some people will actually be 10, 20 years younger than the average person at that same age. And it's known that those people who are younger biologically are healthier and longer lived than those who are already accelerated. So in other words, I could take a blood sample from you today, and then in a couple of days I could tell you when you're likely to die with quite a high accuracy. Now that's important because first of all, it can tell you if you need to act and if you should really pay attention to your lifestyle or to your genes. But it also allows us to quickly establish if we can slow down or reverse the aging process, both in mice and in humans. And there's already some evidence that we can not just slow down this clock, but reverse it, both in mice, as I'll show you. And recently, Stephen Horvath published that he was able to give a cocktail of molecules to some people and reverse this clock by a little bit, just by 2 years. But it's a good starting point. So this is a very important development. We can accurately measure age. So what about reset? Wouldn't it be great if we could take a pill or have an injection that would remind our tissues and our organs how to be young again? Skin would heal, wrinkles could go away, and diseases might be reversed. This is the equivalent of polishing that disc so that we could get back the youthful information. So how could we possibly do this? Basically, what I'm saying is that there's a backup copy of the youthful epigenome in the body, and that we could perhaps use it to reset the cell. Now, we didn't know until, well, really just this year in our lab whether this was possible. But we were hoping it was possible based on a couple of observations. First of all, you can take a tadpole. Was it tadpole, or I think it was a tadpole cell, and put it into an egg of a frog and regenerate a new organism. John Gurdon discovered that and won the Nobel Prize for it. The other thing that we can do these days is clone animals. Dolly the sheep was the first, but now there are many different species that have been cloned, and they live, though you may not realize it, these animals live healthy, normal lifespans, even though Dolly was an aberration. What does that say? That says that probably there's a reset switch. You can start life from scratch. But can we do this in a living organism? Can we take a mouse and reset it? Can we take a human and reset the body to be young? That's what we're working on. The equivalent would be to take our DNA that's now lost all of its beautiful organization and put it back to this structure. So we started working on this really about 4 or 5 years ago, and we had a breakthrough in the last couple of years. A student of mine found a combination of genes that could actually do this. A combination of genes that are normally only switched on in very young embryos and babies. So I mentioned already that we can generate new life from nuclear cloning, and we can generate new animals. What we can also do is take adult cells and turn them into stem cells. This is called reprogramming. We also know that there are other species, such as this marine organism called Hydra, that can regenerate 2 individuals from one. There are some species, salamanders, axolotls. You can even cut the tip off a mouse's finger, it'll grow back. And in our bodies, if you cut out a large part of the liver, it will grow back. These are all signs that it's really possible to do amazing things with our cells in terms of aging and regeneration. So I mentioned John Gurdon won the Nobel Prize for his tadpole experiment. The man on the right, Shinya Yamanaka, a very famous professor from Japan, here having just won the Nobel Prize in Sweden, what he won the prize for was showing that you could take skin cells from adults, or really any cell type you wanted from an adult, and make it into what's called a pluripotent stem cell, a cell that could be turned into any other cell type, essentially erasing the cell's identity. So the cell was no longer a skin cell, it could become anything. And the way he did that was to take a normal cell and in the dish, not in an animal, but in a dish, put in a combination of 4 genes that would normally be switched on in embryos, in eggs when they're fertilized. And he got these stem cells that now are used in medicine, put into people trying to improve the brain function and other organs. These genes encode what are called transcription factors, which are really just gene regulators that bind to genes and turn them on and off. So this was certainly worthy of a Nobel Prize, but the question that we had was, can you use some of these early, young, embryonic genes to reverse aging without causing them to become stem cells? The last thing we want is to create stem cells in the body, because if a cell doesn't have any identity, it'll be a tumor. It can grow. And in fact, if you turn on all 4 of these genes in a mouse, when it's older, it will develop cancer. And in some cases, will kill the animal, other labs have shown. So this was a very risky experiment. But what we did was we looked in cells at turning on some of these genes and found a unique combination that worked to reverse the age of cells halfway, but not all the way. The way we did this was we took a virus, not a dangerous virus. These are domesticated viruses that are used right now for gene therapy in people, for example, to repair genetic defects in the eye to restore their vision. And they're called AAVs. So we packaged up some genes into this AAV and tested if it could reverse the age of cells. Now we chose neurons, in this case in the back of the eye, of a mouse's eye. We wanted to see, could we make those nerves healthier? Now in a young in a young mouse, these nerves will grow back. The video unfortunately is not playing, but this would be a nerve that grows in the dish. And this one doesn't grow. If you damage an adult nerve, it will not grow back. I think we all know that. If you damage your spine or you damage your optic nerve or your brain, it's not going to miraculously grow back. But it does if it's very young. So we thought if we could reprogram these nerves in the eye to be very young, we might get improved repair and maybe even vision back again from those mice. Now this was an optic nerve where it was these nerves are not healthy. If they're healthy, they glow orange, but here you can see that most of these optic nerves in the mouse have died off. Now if we apply 3 of the Yamanaka genes that I told you about, called O, S, and K, and we turn them on for 3 weeks, those nerves grow back. You can see a lot of them are preserved. The eye is here, the brain is on the left, and they grow back. This has really never been seen to this extent, and certainly not seen when the treatment was given after cells had already been damaged. And we call this epigenetic reprogramming. And these nerves, when they were damaged, they aged prematurely and died, perhaps of old age, within a matter of weeks. And here, these nerves were young again. So we can also test vision. In this experiment, we're testing glaucoma. You might have relatives who have glaucoma. It's pressure in the eye. It's one of the largest causes of blindness on the planet. And if you lose your vision because of pressure in the eye, you will never get it back. There's nothing you can do except some medicines that slightly slow down the progression of the disease. But we asked, if we have a mouse that has lost its vision from pressure, can we restore that vision? And we can test this a number of ways. We can look at electrical signals in the eye. But another way we do it is to look at vision. And here's an experiment where the mouse is looking at moving lines. The lines are moving like this. And if the mouse cannot see, it will just sit there. But if it can see, it will move its head like this. And we can track that with a camera. And this experiment here, we just need to focus on the red versus the blue. And what you can see is with our reprogramming, we get back the youth in the cells, and we also get back some vision that's been lost. So this is the first time that anyone's been able to restore vision in blind, or at least mice that have lost their vision. But then we did something interesting. We teamed up with another lab at Harvard that works on vision, and we took just regular old mice that were in the lab and living normally, and we put them in front of the screen. You can ignore, this is just the mouse being a bit nervous on a platform down here, but you can see that this mouse is not moving its head. This mouse cannot see because it's old. It's about 1 year old, and this type of mouse loses its vision rapidly due to old age. What we wanted to do was to deliver our 3-gene combination with the virus into the eye. It's very simple, actually. It's not difficult. Doctors do this all the time, give a single injection into people's eye who have, say, macular degeneration, and it's painless and it's very quick. We did the same for the mouse, didn't feel it, and 3 weeks later, after we turned on these epigenetic reprogramming factors, we got this result. We had old mice that could see. You can tell that this mouse is now able to see again. And in fact, these old mice, on average, could see just as well as the young mice could, which to me is really quite incredible that we can now restore vision in blind mice. My student at the time, Wancheng Lu is his name. He's now a postdoctoral researcher, a PhD, a doctor. He took the cells out of the back of the eye and measured whether they were younger. And in fact, it turns out that they were much younger based on this clock. And not only that, this symphony I was referring to, of which genes are played, it came back to be a beautiful youthful symphony of genes. The genes that went off with aging came back on to the right level, and genes that went on during aging were shut off again. In other words, we had restored the nerve cells' identity. The nerve cells woke up from their sleep in the old mice to function as though they were young again. In fact, literally, they were young again. We actually know that this clock is important, or at least the methylation, those DNA changes, are important, because if we block the eye from removing those chemicals, the vision was not restored. So this clock that I'm telling you about, those chemicals seem to be important for the aging process and the reset switch, which we didn't know until we discovered this recently. One way to think of this actually is, so if you have a clock on the wall, if you turn the hands of the clock backwards, nothing's going to change because the clock is just a clock. But what this experiment is telling us is that this is a very special clock. If you move the hands backward, time goes backward, age goes backwards. Yeah, so I just want to talk a little bit about what we can do in our daily lives to give ourselves the maximum possible health in old age. And we know that how we live our lives has a big impact on us. In fact, most of our longevity and health, 80% in fact, is determined by our epigenome, not by our DNA. And we can change our epigenome by how we live. So what can you do now? Well, if you're getting sufficient food, if you're eating 3 regular meals a day, breakfast, lunch, and dinner, or even eating snacks in between, we know that that's not going to give the best chance of longevity. And certainly overeating will be even worse. So what I do is I like to skip at least 1 meal a day. We have enough food in our household, and our meals tend to be large to begin with. And I try not to snack. I like to have tea and coffee if I get a little bit hungry, and you do get used to it. Now, you may not want to skip breakfast, you may want to skip lunch, you may want to skip dinner. It's different for every individual. I also just want to point out that if you're young, if you're a teenager or in your 20s, this probably isn't for you. I'm talking more about people who are middle-aged, whose metabolism has slowed down, and we certainly don't want to suggest malnutrition or starvation as any way to improve health. That's the last thing that we want to talk about or hint that could be helpful. This is more about being, at least some part of the day a little bit hungry. And we've known this for thousands of years, that this actually is helpful. What we didn't realize was that what happens when you're hungry is that these longevity defenses, these longevity factors that I'm describing to you and others get turned on. If we sit around all day, if we are always eating or not feeling hungry, our bodies will relax and they won't fight aging. So this is one way to turn on those defenses against aging. Do a bit of exercise. Lose your breath a few times a week if you can. If you don't move, get up, walk at least. Do some weightlifting as well. Once a week at least I try to lift something. I do some wrestling with our son as well. That helps. I talk about hip hinge exercise. This is bending your hips and lifting weights. It's very important to particularly keep your hips strong from walking and bending, because just in the US, every 19 seconds somebody falls over and breaks a bone. And this is just as bad as getting cancer in terms of survival. You might want to talk to your doctor and get tested to see what's working in your body and try to counteract that. You can also have devices on your wrist that can tell you how you're doing. And I do that because, I don't take blood tests a lot, but I do try to monitor to see how I'm doing, to see if things are working. Sleep well, reduce stress, this is all important stuff. Sleep is actually intimately tied to these longevity genes that control the pace of aging. And here's one you may not have heard. You want to eat plants that have been stressed. What do I mean by that? Well, red wine is an example of a foodstuff that's made from grapes that have been stressed. You probably want to eat plants that have been grown outdoors or dehydrated. And farmers actually know that if you stress plants before picking, such as orange trees, you can put a nail into the stem and it will produce better fruit. The reason is that plants produce their own molecules to improve survival and longevity. And we believe, and we've got a lot of evidence, that when you eat those molecules from the food, you mimic exercise and you mimic fasting and hunger. We call this xenohormesis. So I've given you a lot to think about today. You can read about the new discovery online at nature.com. It's out there right now. I've tweeted about it. There's a free copy if you want to download it if you don't have a subscription to Nature magazine. And in my book Lifespan, I talk about not just why we age, but what we can do about it in much more detail than I've told you about today, including what I do and my family does. And my father is 81 years old with no diseases, in perfect health, and he's literally stronger and fitter and arguably happier than I am. So it's not a clinical trial, but he is a beacon of hope for all of us.